In the first part, we have seen about the features of cell. What all things are there in the cell we have seen. It consists of plasma membrane, then cytoplasm, then nucleus. We know that cell is able to perform its function by itself. It is because of the presence of various cell organelles. The cell organelles are present in cytoplasm. We all know that cytoplasm is divided into three: cytosis as well as cell organelles. All the cytosis is the aqueous medium present in the cytoplasm, as well as some organelles are seen. They will be floating over these. They are called cell organelles. I have told you. Now we are going in detail about the cell organelles. Can we go? The cell organelles which we are seeing is endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids and vacuoles. What are they? How are they functioning? And how the cell is performing its function because of these cell organelles we are going to see. Actually, not only these, there are much more cell organelles are there, but these are the important one, which are, that's why we are discussing about it. Okay, we have already seen that we can able to see a cell when we are looking in microscope, isn't it? Like looking through the microscope. But if we are just seeing through a normal microscope, we can just see the cell and a nucleus also can be seen. That's all. But if we are looking through an electron microscope, we are able to see these organelles clearly. Okay, that much small they are, but they will perform their functions quickly and smoothly, which make the cell to be alive. We are moving for the first one, that is ER, endoplasmic reticulum. What is that? Actually, it is a membrane-bound structure which is present in along with the nucleus is this endoplasmic reticulum. Network of membranes can be seen. Okay, this endoplasmic reticulum is divided into two. That is smooth endoplasmic reticulum as well as rough endoplasmic reticulum. That is SER and RER. I have mentioned. Smooth endoplasmic reticulum means above this endoplasmic reticulum there is no presence of a certain substance, so it will be so smooth. Okay, what is that substance? I will tell you. Okay, ma. The endoplasmic reticulum is so smooth. It helps for what, ma? It helps for the synthesis of lipids, steroids, and fat molecules. What, ma? Lipids, steroids, and fat molecules can be synthesized in what? This smooth endoplasmic reticulum. Okay, is it clear? Now we are moving for the rough endoplasmic reticulum. By the name itself, it is clear that endoplasmic reticulum is not smooth. Something is there over it to make it rough. Isn't it? Have you seen the sandpaper? Sandpaper and normal paper is different. Why? So normal paper is smooth while sandpaper is having some sands over it, and that's why it is so rough. Like that, this endoplasmic reticulum, something is kept on the top. That is ribosomes. What is that, Kana? It is ribosomes. These ribosomes are kept on the top of this endoplasmic reticulum. So it makes the endoplasmic reticulum so rough. Okay. When the ribosomes are attached, wherever you have to have a memory in you, that is, whenever you are hearing ribosomes, you should understand that if ribosome is there, protein will be synthesized. What is that? If ribosome is there, what will happen? Protein synthesis is happening. So this RER, that is rough endoplasmic reticulum, will produce what, ma? Protein. So we have in the last class itself we have told that a factory is there. We are getting inside only with the help of what, ma? Plasma membrane. Plasma membrane helps for the entrance. When we are getting inside, there is a production section. Endoplasmic reticulum is doing some of the production. What are they? This endoplasmic reticulum making fat molecules, steroids, lipids, then protein. All these are synthesized by what? This endoplasmic reticulum. Is that clear for you? Okay. Then what are some other functions of it? Not only that, transportation of material between cytoplasm as well as nucleus is done by this endoplasmic reticulum. The things which are needed for the nucleus, this endoplasmic reticulum will transport it to nucleus. Okay, from the nucleus, if something has to be kept gone for cytoplasm, this endoplasmic reticulum also help for that. Transportation of materials between the cytoplasm and nucleus is done by what, ma? This endoplasmic reticulum. Okay. Then it provides space for biochemical activities. For some biochemical activities to be done, it provides space. Okay, that's what you have to keep in your mind. Then the third one I told you, fats, steroids, then proteins, all these are synthesized by ER. Then the fourth and important point is it detoxifies. It detoxifies the poisonous substances. If there is any poisonous substance entering into it, it will detoxify. ER will detoxify it. Okay. This proteins and lipids, you have heard something. That is proteins and lipids. Something is made of proteins and lipids. Do you remember what is that? There is an entry. What is that? Plasma membrane. Plasma membrane, I told you that time itself. Plasma membrane is made of what, ma? Proteins and lipids. Now you are able to understand from where this protein and lipids are secreted for making this plasma membrane, isn't it? So endoplasmic reticulum is responsible for that also. Okay. This is called membrane biogenesis. It is called what? The proteins and lipids which are useful for making the cell membrane is called what, ma? It is called membrane biogenesis. Now ER is smooth for you, isn't it? We are going for the next one. The second important thing is Golgi apparatus. Golgi bodies or Golgi apparatus we can say. Okay, ma? The name is not that much familiar for you. Yes, it is Golgi apparatus. It's easy for you to learn. Okay. Actually, from the picture itself you can see it is the system of tubules and the membrane bound vesicles can be seen. Membrane bound vesicles can be seen. How the structure is, if you are seeing, you can understand how the Golgi bodies or Golgi apparatus are. It will be along with ER. What is ER? Endoplasmic reticulum. It will be side by side with ER. What is its main function, you know? We know that ER is the production. Production is doing in ER, isn't it? Lipids, proteins are produced. Moreover, that steroids, fat molecules are produced by ER. This Golgi bodies or Golgi apparatus will do the packing section. It is having the packing section. So, whatever the proteins and lipids which are synthesized in ER will be packed by what, ma? Golgi apparatus. Moreover, this proteins and lipids which are synthesized by ER packed by Golgi bodies can be transported between the cells or from the cell to outside also. It helps for the transportation of the synthesized material. Okay, but how it is how they are transporting? Only by doing the well package. Okay, if in a chocolate factory, chocolates are made by ER, isn't it? Endoplasmic reticulum, and this Golgi bodies will pack it. This wait to say chocolate, how it is packed, no? In fine paper, like that it will be packed in so beautiful papers and all. After that, from the factory, it will be moving out. Okay, it is done by what, ma? Golgi apparatus. So, if the Golgi apparatus is not there, what will happen? It's also responsible. It's very, very important thing. If the Golgi apparatus is not there, this packaging and dispatching will be stopped. From the cell to outside will be stopped. So, it is very, very important. Then it also helps for the formation of middle lamella. Golgi apparatus is responsible for the formation of middle lamella. Now, I think you are clear with Golgi apparatus. Now, we are moving to see lysosomes. What are lysosomes? Now, we are moving to see yet another important thing, that is lysosomes. Lysosomes, they are very tiny. They are like so granules. Why then this small thing are having that much importance? Actually, it is very powerful. Why? You know, because it helps for digestion. For the digestion purpose, these lysosomes are used. Actually, this is a small granule-like structure. It contains digestive enzymes. Actually, if any foreign particles are attacking to the cell, if it entered via plasma membrane and if it is going to attack the cell, This lysosome will secrete this digestive enzyme and it helps for digesting this foreign particles so that the cell will be escaped and so it will be protected. This is the importance of lysosomes. This lysosome is having single membrane covering. Not only for the digestion purpose, it also helps for the disposal of the waste. This lysosome is having yet another nickname. Do you know what it is? It is called suicide bag. Why it is called suicide bag? If any cell is damaged, if the cell is not able to work, anything has happened to the cell, what will happen? This lysosome will burst, and what will happen when it is bursting? This enzyme will be secreted. What will the enzyme do? The enzyme will digest the cell itself. Okay, so it is called suicide means what? By killing oneself. If one is killing himself or herself, we are saying it as suicide, isn't it? So this lysosome able to kill the cell itself. So it is called suicide bags. Normally it will not function as a suicide bags, but sometimes some damage has been caused for the cell. For the safety purpose, the cell, this lysosome will burst off and the enzyme will be secreted and it will help for the digestion of this cell, entire cell itself. That's why it's called suicide bag. It may be asked for your question paper, in the question paper. Now we are moving for the important part, that is mitochondria. Name is so to say we like it, no? Mitochondria. You can give to your children or someone. Mitochondria, mitochondria come here, mitochondria sit there, mito baby come here, you can say, right? Mitochondria, the name itself is so powerful. Mitochondria is the powerhouse of the cell. It is the storage battery or it is the powerhouse of the cell. That's why I told you to give names as mitochondria. Powerhouse, powerful it is, right? That's fine. Okay, why it is powerful? Why it is powerhouse? We are saying it as powerhouse because it is the one who is having energy. They can produce energy and they can give energy to the other cell organelles for functioning smoothly. That's why the mitochondria is called what, ma'am? It is called the powerhouse of the cell. You should remember one thing, that is mitochondria will be seen in eukaryotic cell, and moreover it is double layered membrane bounded organ thing. Okay, double stranded membrane bound is there for nucleus also. For mitochondria also double layer is there. This membrane coating is double layered. Okay, ma'am? This mitochondria is the rod shape. It will be in which shape? Rod shape. From the picture itself, it is clear for you that mitochondria is rod shape. And it is having double membrane coating. The outer coating is made of protein. Outer coating is made of protein, while the inner thing we can see some, can you see from the picture itself? It is some strands can be seen. We are saying it as cristia. We are saying it as cristia. Okay, ma'am? Now, what are the functions of mitochondria? We are going to see. What is the function of mitochondria? It helps to produce and store energy. It is the one who can produce energy and can store energy in the form of ATP, adipose triphosphate. You have to learn it. Okay, ma'am? In the form of ATP, it can produce and it can store energy. And whenever or any cell organelles need this energy, it will give that energy so that they can function much more better. So that's why it is called the powerhouse. Not only that, it is having its own DNA. Mitochondria is having its own DNA and it is having ribosomes. What are you understanding by the name ribosomes? Which means mitochondria is able to synthesize protein by itself. It need not depend on any other place. Give me some protein, no need of it. Mitochondria is having ribosomes, so it can synthesize its protein. You know that mitochondria have outer covering made of protein for the synthesis and all. I am having my own things, isn't it? Ribosomes are there, so I can prepare. Mitochondria will say. So it is the one who gives energy, and that's why it is called the powerhouse of the cell. Now yet another important part is plastids. Plastic noma, it is plastid. Okay, plastids, where it is found? It is not found in animal cell, but it is found in plant cell only. Okay. What is the thing which the plants are having but animals are not having? Green color has for photosynthesis. All these are related to this plastid. Did you get ma? Okay. So plastid means something is helping for photosynthesis. So the plastid is divided into three main parts, that is leucoplast, chromoplast and chloroplast. There is no handiplast. Okay. Three plasts, what are they, ma? Leucoplast, chromoplast, chloroplast. Leucoplast is white in color. It is found in the down part. Consider leaf. Okay, we are considering leaf and we are saying in the leaf, this white color can be seen where, ma? Can we see at the top portion of the leaf? No, it will be seen in the base of the leaf, right? So the base part is having leucoplast. Okay. Then the second is chromoplast. Chromoplast is colorful, red, brown color it will be in. Okay, this red and brown color where we can see in leaf. It can be seen in the top itself. If we are imagining this red colored or colorful plant, colored leafed plant, the red color can be seen, no? Brown color can be seen. Like that, this colored plants will be having this red and brown color. Okay, this chromoplast is having the red and brown color now. Now we are moving for chloroplast. We have heard about the word chlorophyll. Is found in chloroplast. We have learned from 4th standard itself, right? This chloroplast is green in color and it is found in the aerial part, means in the top part of the leaf. This chloroplast is seen. Is it clear for you? Okay. Now, this chromoplast and chloroplast helps for what? Photosynthesis. Because chloroplast contain chlorophyll help for photosynthesis. We have already learned it. So, this chloroplast and chromoplast help for photosynthesis, while the leucoplast, what will it do, you know? It will store the things. What are things? Starch, oil, protein, some protein molecules, whatever is synthesized will be stored in leucoplast. Is it clear now? Okay, this is about the plastids. This is that much about the plastid. Only one thing you have to keep in mind, that is plastid will be seen only in plant cells. Is it clear? Now we are moving for vacuoles. What are vacuoles? Vacuoles are, from the name itself, it's a membrane bound region which is present in both animal cell as well as in plant cell. In the animal cell, the vacuoles are small in size, while in the plant cell, the vacuoles are larger sized. For what purpose the vacuoles are? It is the storage house. In the factories, the things, chocolates are, oh wow, we forgot our factory, right? Production has been gone, then packing section has been gone, storing house also is there, isn't it? The storage house is vacuoles. The water, amino acids, and is there anything more? Yes, sugar, protein, all these are stored in this vacuoles. Have you heard about amoeba? In amoeba, vacuoles, large vacuole is there in amoeba. The food items also will be stored in this vacuole. Okay, ma? Then we have to see for what purpose this vacuoles are used. It is for the storage purpose. Not only that, if excess water is there, it will be expelled out by what? Vacuoles. We have already told the water is stored. If the excess water is there, what will happen? It will expel this excess water out. This is also done by vacuoles. Now we have finished almost everything. Endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastid, vacuoles, all these. Now we reach the last part of the next chapter, that is the difference between plant cell and animal cell. Plant is also having cell, animal is also having cell, but there are much more difference in the plant cell and the animal cell. Can you say without my help anything? Okay. We can see plant cell is having cell wall. Cell wall can be seen in plant cell. Is there anything for the animal cell? No cell wall, only plasma membrane is there. Remember the previous things. Okay. Second is this chloroplast is present for what? It is seen only in plant cell but not in animal cell. And the third point is centrioles. Centrioles can be seen in plant the animal cell but not in plant cell. Okay. Then the vacuoles we have discussed now only. Vacuoles are larger in the case of plant cell but smaller in the case of animal cell. Then the nucleus. Nucleus in the plant cell is in a side because of this large vacuole. Vacuole will occupy much more space, so the nucleus will have to go inside. But in the case of animal cell, the vacuole is small, so the nucleus can be in the center. These are the main difference between plant cell and animal cell. Now, an important thing that is about ribosomes. I told you already that ribosome is also called protein factories. It is also called what, ma? Protein factories. That means it is the one which is able to synthesize protein. Do you know it is a minute particles which contains RNA. RNA is present only in the main ribosomes. Okay, ma? Then it helps for the synthesis of protein. Not only that, the synthesized protein is transported to endoplasmic reticulum ER by this ribosomes only. We have seen endoplasmic reticulum are of two types, smooth one and the rough one. Rough one will be having ribosomes, and this ribosomes synthesize what, ma? Protein, and it will transfer it to endoplasmic reticulum. From there, it will be packed to packing section Golgi Apparatus. Do you remember? All these has been revised again, right? Now we are winding the chapter. We are here to learn about the important thing, that cell division. How do the cell divides? We know cell divides and develops to form various organs, organ system and organism, isn't it? So divisions of cell is very important. Two types of cell divisions are there, that is mitosis and meiosis. What is the difference between mitosis and meiosis cell division? Mitosis can be seen for all for the organism. Normally the cell divides by mitosis way. That is, consider this as the mother cell. It divides to form two daughter cells. Here 1, 2, 3, 4. Here also 1, 2, 3, 4, 1, 2, 3, 4. That is number of chromosomes they are representing. The number of chromosomes remains same when they are dividing. Identical division, identical twins will be formed, identical child will be formed, and these identical cells will be having the same number of chromosome as the mother cell is having. Okay, this kind of cell division is called mitotic cell division. Here they have represented the same chromosome number remains the same. In the case of meiosis, the second cell division is meiosis. This meiosis type of cell division will be found only in reproductive cells. For the reproductive purpose, the cells are used. By the time meiotic cell division will be formed, there not only one division, two successive divisions will be formed, result in the formation of 4 cells. Here 2 cells has been generated from a single cell. Here instead of that 4 cells has been generated. 1, 2, 3, 4. But the number of chromosomes will be exactly half of that of the mother cell. See here 1, 2, 3, 4. Here 1, 2. 1, 2, 1, 2, 1, 2. Which means number of chromosomes will be exactly half of that of the mother cell. This is called mitotic and meiotic cell division, and this is called mitotic cell division. Okay, dears? Now it's question time. Answer these questions. Dash transport material between cytoplasm and nucleus. It's a cell organelle. What is that? Which transport material between cytoplasm and nucleus? Second question. Cell wall is made of dash. Cell wall is made of dash. Third one is movement of water molecule through plasma membrane is called dash. Movement of water molecule from higher concentration to lower concentration through plasma membrane is called dash. Answer this question. Thank you for watching.